Hello and welcome to Biology class. I am Shipra Sharma from ADARSH MANDIR, SHANKAR, Mount Abu. For class 12, today we will start a new chapter, that is Biotechnology, Principles and Process. In this chapter, first we have to know what is biotechnology. So biotechnology is a technology using the living organism to produce which are useful for us, like enzymes, vaccine, antibiotic, etc. The next is genetic engineering. It's a principle of biotechnology. What is genetic engineering? Genetic engineering is the technique to alter genetic material, that is DNA or RNA. In some viruses, the genetic material is DNA, and in other viruses, the genetic material is RNA. So, by this genetic engineering, the technique to alter the genetic material to introduce these into the host organism. Generally, the host organism are the bacterial cells like E. coli, and thus change the phenotype. Phenotype is external appearance. Phenotype is the expression of genotype. If the genes, the sequence of genes will be changed, then phenotype is automatically changed. So, it is the basis of genetic engineering. And what are the use of biotechnology? Biotechnology is useful in the production of antibiotics, vaccines and enzymes. What is antibiotic? Antibiotic are the drugs which are used to destroy the pathogens. Pathogens means causal organism of specific disease. So antibiotics are the drugs which are made by the use of this biotechnology, and these drugs can destroy the bacteria, viruses and protozoans, all types of microbes. They can cause the disease. And the next one is vaccines. What are vaccines? Vaccines is the dilute form of causal organism. Vaccines are the dilute form of causal organism which is injected into our body to develop the natural antibodies in our body. Okay? So, vaccines are very important, like for TB. TB is the tuberculosis. It's a very dangerous disease. But BCG is the vaccine of TB. So, what is BCG? BCG is the causal organism of tuberculosis bacteria, and it injected into the body. So, what happens in the body? In our body, the WBC are present in the blood, and WBC form the antibodies against the causal organism of TB. Okay, so this time the vaccines will act, and some enzymes are also being formed by the biotechnology, like insulin. Insulin is a type of hormone, but it's basically the enzyme. It's a protein. Okay, so what are protein? Protein are the polymers of amino acid, and amino acids are binded by the peptide bond. So they form the enzymes. So many types of enzymes can be formed by this technique. Now, what are the tools of recombinant DNA technology? The most important is the restriction enzymes. Restriction enzymes are also known as nuclear scissors, and they can cut the DNA at specific sites. So these restriction enzymes are of two types, that is endonuclease and exonuclease. So what is the difference between endonuclease and exonuclease? Exonuclease remove the nucleotides from the ends of the DNA fragment. Okay. So what are the nucleotides? Nucleotides is the molecule which is formed by one nitrogenous basis, one pentose sugar and one phosphate. So what are DNA? DNA is the polynucleotide strands. So exonuclease can cut these nucleotides from both the ends, and the endonuclease, it can cut the DNA at a specific position inside the DNA segment. Clear? Now, what is palindromes? Palindromes, these are groups of letters. Here the letters are the nitrogenous bases, okay, that form the same words where we read from both the sides, forward and backward, like Malayalam. Malayalam means M-A-L-A-Y-A-L-A-M. write backward, what happens? From here, M, A, L, A, Y, A, L, A, M. Now you can read from both the sides, forward and backward. Both are same, both are the real. So these are known as the palindromes. Okay, these are sequences are known as palindromes. You can see here, this is the sequence. It is 5' to 3' strand of DNA, and in this DNA, this is the sequence of nucleotide, that is GAATTC. And what will be the palindromic sequence of this DNA fragment? The palindromic sequence will be from 3' to 5', that is C, T, T, A, G. Clear? So from both the side, here G, G, C, C, T, T, and A. These are the palindromic sequence. Now, how they work? For example, this is the vector DNA. This is the vector DNA, and here is the nucleotide sequences. We have shown these nucleotide sequences, and this is the foreign DNA. Foreign DNA means the DNA fragment which is isolated from the other organism. So this is the foreign DNA, and what happens? Just first we isolate the sequence. What are sequence? Palindromic sequence. Both the sequences are same. You have seen GAATTC. Here also GAATTC. So in both the DNAs, vector DNA and foreign DNA, both the sequences are same. So these are known as palindromic sequence. So from the place where palindromic sequence are present, that restriction enzyme will work and cut the sequences. And you can see here this sequence is cut, and here this sequence is cut. And these are known as the sticky ends. This is the sticky end of vector DNA, and this is the sticky end of foreign DNA. You can see here. And now with the help of DNA ligase is the enzyme, it can join the sequences. This is the vector DNA and this is the foreign DNA. So vector and foreign DNA segments can be joined with the help of DNA ligase. Now here we can see the DNA recombinant technology. Here is the DNA recombinant technology. We have seen this is the foreign DNA. Foreign DNA, that is the specific DNA which is isolated from the other organism, and this is the vector DNA. It is a plasmid. It is a cellular DNA which is present in E. coli or other bacteria. Okay, we take the bacteria for this technology because bacteria, the reproductive rate in bacteria is very high. In every 20 minutes it can replicate. That's why we take the bacteria. So this is the bacteria E. coli's plasmid cellular DNA, and with the help of restriction enzymes. Which are the restriction enzymes? These are the nucleocases, endonuclease and exonuclease. So we inject the restriction enzyme inside these DNAs. Then what happens? You can see here, this is the single strand, and now it is divided into 3 fragments. So it is cutted by the help of restriction enzyme. And here also you can see this is the plasmid, and this plasmid is cut from this side. So it is the use of restriction enzyme. Now what happens? After this, the use of restriction enzyme, we use the DNA ligases. What is the function of DNA ligases? DNA ligases joins the DNA fragments. So what happens? This foreign DNA is joined into this plasmid. So here you can see this is the part of foreign DNA and this is the plasmid. So now this plasmid is joined with the foreign DNA, specific DNA, and it is present inside the bacteria, E. coli. So what happens? Every 20 minutes it can replicate and reach up to the next generation, because DNA is the hereditary material. So by this process it multiplies and number of DNAs are increased then what happens? Then we just separate the DNA material from these E. coli and use it for the antibiotic production or the enzyme productions. Okay, thank you.